Okay, welcome back. This is Enzymes and Medicines Part 2. There's a name that a word you might not have heard of before, and it's Zwitter ion. It comes from the German word for hermaphrodite. It's also known as an inner salt or dipolar ion. And here we have Zwitter ion. Now, you might recognize it as being an amino acid, except the amine part is protonated. It's got a positive charge and an extra hydrogen. And the carboxylic acid group, the carboxyl group, has lost its proton. It's deprotonated and it has a negative charge. So there's a positive and a negative charge on this amino acid. That makes it a Zwitter ion at pHs around neutral. So where the concentration of hydrogen ion is similar to the concentration of hydroxide ions. At very high concentrations of hydrogen ion, or low pH, we have a cation form. So the carboxyl group is complete, but we have a protonated amino group. And at very high pH, or high concentration of hydroxide ions, we have a deprotonated carboxyl group and a regular amine group. So amino acids in solution form ions very easily. And the presence of the ionic groups is very important for bonding and the structure of proteins. The Zwitter ion has no overall charge. And we'll come back to this during this lesson. So here we're looking at Enzymes and Medicines Part 2. In this lesson, at the effect of pH on enzyme function and medicinal enzyme inhibitors as well. So back to where we started in this lesson. So we've got our Zwitter ion here. It's got a positive and a negative charge. Overall, it's neutral. But we see that the effect of pH on a system that contains amino acids or proteins, the pH is very important because at low pH, we have a protonated cation form of an amino acid, and at very high pH, we have a deprotonated carboxyl group and a regular amino group. So amino acids change their structure with pH, and that's going to have a big impact on the peptides, polypeptides, proteins that they're part of. So in an acidic solution, pH approximately 1, so highly acidic, the amino group on an amino acid will protonate. In other words, it will gain an extra H+, so the NH2 becomes NH3+, and we put the plus on the nitrogen. In a basic solution of pH approximately 13, very high pH, the carboxyl group will deprotonate, forming an anion. So here's our anion part, the carboxylate ion. And in a solution of around neutral pH, and it does depend on the amino acids involved, both the amino group is protonated and the carboxyl group is deprotonated, forming a Zwitter ion. Now, that's as far as you need to know about the effect of pH in VCE. If you go on to do university chemistry, you'll learn more about specific pHs for specific amino acids. What you do need to know is what we mean by an acidic amino acid and a basic amino acid. So amino acids have all got a carboxyl group, which is an acidic part anyway, and an amino group, which is basic. So all two amino acids, all alpha amino acids have that. What varies between the amino acids is the side chain, which is often given the symbols Z or R. And in this case, the side chain contains a carboxyl group. So if we lose 2 protons, one from the backbone carboxyl group and one from the side chain carboxyl group, then we've got a -2 ion, and that can be involved in ionic bonding. So acidic amino acids are aspartic acid and glutamic acid. They have carboxyl groups as part of their side chain. Basic amino acids, on the other hand, have an amino group in their side chain. They've still got a carboxyl as part of the amino acid structure and an amine, but they've got an additional amine group as part of their side chain. So in a highly acidic solution, so perhaps pH 1, both the amino groups, the part of the general amino acid structure and the part from the side chain, both protonate, forming a +2 ion, which again can be involved in ionic bonding. Now, the effect of pH on enzyme function. So we have our substrate, that's what's going to change shape or change its structure during the catalyzed reaction. So here we have a model of an enzyme and the substrate. The substrate has a particular shape, which, like a lock and key, fits into the active site and forms the enzyme substrate complex. And the type of bonding that can occur is going to be really influenced whether or not there are positive or negative charges. So the presence of hydrogen ions or hydroxide ions, in other words, low pH or high pH, affects the secondary, tertiary and quaternary structures of enzymes. So proteins are very sensitive to pH changes and can easily denature. Now, enzymes have different optimal pHs. Pepsin that's found in the stomach has an optimal pH of around 2.5, whereas catalase, which is found in the liver, has an optimal pH of 9. So it depends on the location of an enzyme in our body what its optimal pH is. So salivary amylase, which is an enzyme that catalyzes the breakdown of starch, starts when we start chewing food in the mouth, because saliva has a pretty neutral pH of 6.2 to 7.6. When that amylase hits the stomach, the pH drops significantly, round about pH 2, which is the optimal pH for pepsin, but the salivary amylase will be denatured in the stomach. So it will no longer have that active site. So after food passes through the stomach, which is a low pH, then the food passes into the small intestine, where the pH is back around the neutral region. And pancreatic amylase is secreted into the small intestine to continue the starch digestion because the salivary amylase has already been denatured. Note that blood has a very small pH range for healthy living, between 7.35 and 7.45. So various enzymes operate at different optimal pHs and can be easily denatured outside that pH range. So let's have a look at a worked example. Draw the chemical structure of the dipeptide cis-asp. Cis stands for cysteine, which you need to look at in the data book, and asp, aspartic acid, at high pH. Okay, so what we do is we start off with cis, the left-hand side, then we get carboxyl group from the cysteine and the amine group from the aspartic acid. We join them together. We also lose a water molecule. We form our amide link, and at high pH, we're going to deprotonate the carboxyl groups. So we have COO minus, COO minus. So correctly drawing the amide group is 1 mark, and correctly drawing the rest of the structure, including deprotonating both carboxyl groups, is also worth 1 mark. Okay, now we're going to talk about medicinal enzyme inhibitors. So a competitive inhibitor can bind to an enzyme's active site, which makes them very useful as medicines because they can stop an enzyme from working inside the human body. For example, angiotensin-converting enzyme inhibitors, or ACEIs, are used to treat high blood pressure, heart failure, diabetes, and kidney disease. And normally, without the presence of the enzyme inhibitor, we would have angiotensin 1 binding to angiotensin converting enzyme to form angiotensin 2, which causes smooth muscle to contract. You wouldn't need to know the details of this for the chemistry exam. But instead of angiotensin 1 binding to the enzyme, we can have a similar shape, an ACE inhibitor that binds to the enzyme and stops the formation of angiotensin 2, which stops the smooth muscle from contracting. So by interfering with the normal process, some medicines can be beneficial. So another example is a medication for Alzheimer's disease. It's a competitive reversible enzyme inhibitor that blocks the breakdown of acetylcholine in the body. Acetylcholine, you don't need to know the structure of it, but it has a very similar shape to the medication for Alzheimer's disease, a square and a circle sort of shape, that can mean that instead of the acetylcholine binding to the enzyme inhibitor instead binds, and then the acetylcholine doesn't break down. And that can be beneficial in the treatment or the medication for Alzheimer's disease. Okay, let's have a look at a worked example. Statins are molecules found naturally in oyster mushrooms, so that we've got here. And fluvastatin is a medicine that acts as an enzyme inhibitor. It inhibits the enzyme HMG coenzyme A reductase in the body. Now, you don't need to know anything about that enzyme, but it's required for internal synthesis of blood cholesterol. So it's not just dietary cholesterol, but our bodies actually make their own cholesterol. So identify the type of bonding that occurs between the medication, the fluvastatin, which is in yellow, and the HMG coenzyme A reductase enzyme. And we've got the active parts of the substrate, using the model provided, and explain the effect of the medication on the enzyme. So if we look closely, we can see some hydrogen bonding between the medication and the enzyme that synthesizes cholesterol in the body. According to the model, the medicine binds to the active site of the HMG coenzyme A reductase enzyme through hydrogen bonding. We can clearly see that in the diagram. The enzyme therefore cannot synthesize cholesterol in the body, and that's the mechanism where it lowers blood cholesterol levels. So there's 1 mark for each of those. Okay, let's summarise what we've learned. Amino acids change their structure depending on the pH of the solution that they're in. At neutral pH, amino acids exist as Zwitter ions, with a protonated amino group and a deprotonated carboxyl group, and that overall have no charge. Low pH, the amine groups are protonated. At high pH, the carboxyl groups are deprotonated. Acidic amino acids have a carboxyl group as part of the side chain, the R or the Z group. Basic amino acids have an amino group as part of their side chain, an R or a Z. And enzymes have an optimal pH. Outside of the usual pH range for the particular enzyme, they'll be denatured through changes to the secondary, tertiary and quaternary structures because they're very sensitive to the concentration of hydrogen ions and hydroxide ions. Medicinal enzyme inhibitors. Some medicines work by competitively binding to the active site of body enzymes, preventing their usual mode of action of a particular enzyme. And examples include ACEIs and donazepam.